Good afternoon all. So this is week 12 problem solving session. So this is the fabrication technique for MEMS-based sensor clinical perspective section. I'm Arpita Biswas, a PhD scholar from IIT Jodhpur and electrical engineering department. So this is more details about me. So in this session, we are going to solve some question. So if you have any doubts, you can clear here. So for 2-3 minutes, we are going to start. So let's start the session. This is question number 1. We have microfluidic devices with a channel dimension that can flow 5 microliter per minute of liquids through them. So this flow through the micro channel can be achieved using the first option is the peristaltic pump, second is the centrifugal pump, and third option is the rotary lobe, and fourth option is inline stump. So please go through the question and try to find out the right answer. So what is the right answer? Question number 1, the right answer is option A, peristaltic pump. Okay. So next move toward the question number 12. Modeling a MEMS microheater requires solving dash and dash physics. The first option is electrical and structural physics. The second option is electrical and thermal physics. The third option is structural and thermal physics. And the fourth option is fluid flow and mechanical physics. So what is the right answer? A modeling a MEMS microheater requires solving dash and dash physics. So find out the right answer. So the right option is option B, electrical and thermal physics. Okay. So next move toward the question number 3. Glass wafer to silicon bonding can be done by, first option is the plasma bonding, second the direct wafer bonding, and third option is the anodic bonding, and the fourth option is by using a adhesive. Okay, so what is the right answer? Glass wafer to silicon bonding can be done by plasma bonding, direct wafer bonding, anionic bonding, or by using the adhesive. So go through the question carefully and find out the right answer. So what is the right answer? So what is the right answer? So the right option is option C, anionic bonding. Okay. So next move toward the question number 4. What is the flow modeling ordered for FEM simulation console? So the first option A is make geometry. The second option is post process. The option C is add material. Option D is choose the type of study. And option E is apply physics condition. So what you have to find out the right option. So first option is A, B, E, C, and B. The second option is C, A, D, B, E, and the option C is B, A, C, D, E, and option D is A, C, E, D, B. So go through the question carefully and find out the right answer. So question number 4, the right option is option D, ACEDB. It means first make the geometry, second add the material, and third apply the physics condition, and then option D. It's been choose the type of study, then B. It's been the post process. So the right option is option D. So next move toward the question number 5. How can a perfect bubble-free PDMS be obtained to fabricate a microfluidic system? First option is the periodic degassing, second option is the hot plate, and third option is the oven, and fourth option is the cryo treatment. So go through the question carefully and find out the right option. So what is the right option? Question number 5. The right option is option A, the periodic degassing. So next move toward the question number 6. Evaluate whether the following statement is true or false. So the statement is only predefined multiphysics can be used in console multiphysics. So is this true or false? So the right option is option B. It is false. So next move toward the question number 7. File format used for 3D printing is or are GBR. Second option is the STL. The third option is the XLS. And the fourth option is ZDS. So which one is the right answer? The right option is option B, STL. Next, move toward the question number 8. In the laboratory, cells are grown in an incubator environment with first option is 27 degrees centigrade, 5% CO2 and 95% relative humidity. The second option is 37 degrees centigrade, 5% CO2 and 95% relative humidity. And the third option is 37 degree centigrade with 5% CO2 and 75% humidity. And the fourth option is 27 degrees centigrade, 5% CO2 and 75% relative humidity. So go through the question carefully. In the laboratory, cells are grown in an incubator environment with, so which one is the right option? So the right option is option B, 37 degrees centigrade, 5% CO2 and 95% relative humidity. So next move toward the question number 9. Bonding PDMS to glass can be achieved using first option is adhesive, second is the UV chamber, third option is the RF chamber, and option B is oxygen plasma chamber. So which one is the right option? Bonding of PDMS to the glass can be achieved using adhesive, UV chamber, RF chamber, or oxygen plasma chamber. So the right option is option D, oxygen plasma chamber. So next move to the question number 10. Evaluate whether the following statement is true or false. Cross-linking agent is used along with PDMS elastometer to improve its mechanical strength. So is this statement true or false? So this statement is true. So next move to question number 11. What is the primary function of a peristaltic pump in microfluidic devices? So first option is measure the volume of the liquid. Second, circulate fluid with high precautions at micro level flow rates. And the third option is heat the liquid sample to a specific temperature. And option D is detect the pressure changes in the fluid. So which one is the right option? Sure. So option B is the right option. Circulate the fluid with high precautions at multi-level flow rate. Next, move to question number 12. Which type of fluid sample can be used in a microfluidic device connected to the peristaltic pump? First is the gaseous sample only. Second, the solid particles. Third, the liquid samples such as blood or drugs. And option D is none of the above. So go through the question carefully and find out the right answer. So the right option is option C, liquid samples such as blood and drugs. So next move toward the question number 13. What is the approximate inner diameter of a tube used in the peristaltic pump as mentioned in the lecture? So first option is the 1 millimeter. The second option is 0.12 millimeter. The third option is 2 millimeter or option D is 0.5 millimeter. So go through the question carefully. What is the approximate inner diameter of the tube is used in the peristaltic pump as mentioned in the lecture? So what is the diameter? So the right option is 0.12 millimeter. So the diameter is 0.12 millimeter. So next move to question number 14. What is the what is a key characteristic of microfluidic devices as explained in the lecture? First option is they use the large quantities of liquid. Second, they consist of channel with micro level feature. Third is they are only used for gas samples. And option D is they do not require precise flow control. So what is a key characteristic of a microfluidic devices as explained in the lecture? So the right option is option B. They consist of channel with micron level features. So next, move to the question number 15. How does a peristaltic pump push fluid through the tube? So first option is by heating the fluid and creating pressure differences. Option B is by rotating the cassettes that compresses and release the tube and pushing the fluid forward. And the option C is by using a piston to pump the fluid directly. And option D is by generating a vacuum that pulls the fluid through the tube. So what is the right answer? So the right option is option B, by rotating the cassettes that compress and release the tube, pushing the fluid forward. So next move towards the question number 16. What happens if the flow rate in a microfluidic device is not properly adjusted? First option is the sample will evaporate. Second, the device will heat up. And the option C is leakage may occur at the inlet or outlet port. Or option D is the pump will stop automatically. So the right option is leakage may occur at the inlet or outlet port. So next move to the question number 17. Which of the following is a function of the peristaltic pump interface? First, to adjust the rotation speed of the pump. Second, to display the temperature of the fluid. Third, to monitor the pH level of the sample. And option D is to change the direction of the fluid flow. So the right option is option A, to adjust the rotation speed of the pump. So next move question number 18. What type of sample can be tested using the microfluidic device as discussed in the lecture? So first is the airborne particles, second cells or tissue such as cancer tissue. The option C is only the chemical solution and option D is the heavy metals. So what is the right answer here? What type of sample can be tested using the microfluidic devices as discussed in the lecture? So the right option is option B, cell or tissue or such as cancer tissue. Okay. So next move question number 19. What is the primary purpose of a CO2 incubator in a biology related lab? First option, to measure the size of cells. Second, to provide a control condition for cell growth. And option C is to sterilize the lab equipments. And option D is to observe the cell under a microscope. So what is the right answer? So the right answer is option B, to provide the control condition for the cell growth. So what is the primary purpose of a CO2 incubator in a laboratory, in a biology related lab? So to provide the control environment for cell growth. So next question number 20. Which of the following is not a parameter maintained by a CO2 incubator for the optimal cell growth? So first option is the 37-degree ambient temperature. Next, 5% carbon dioxide concentration. Option C is the 95% relative humidity. And option D is the 100% oxygen concentration. So which one is the right answer? So the right option is option D, 100% oxygen concentration. Okay. So, which of the following is not a parameter maintained by a CO2 incubator for optimal cell growth? Is 100% oxygen concentration, because we have to maintain the 37-degree cabinet ambient temperature, and also 5% carbon dioxide concentration is needed, and also 95% relative humidity is required. So 100% oxygen concentration is the wrong answer. Okay. So our right option is option D. So next move the question number 21. What is the purpose of the 90-degree sterilization features in the CO2 incubator? So first option is to speed up the cell growth. Second option is to maintain the CO2 level. Third option is to kill any bacteria present and prevent the contamination. And option D is to heat the cell culture media. So which one is the right answer? Okay, go through the question. I'm coming back in 1 minute. So what is the right answer? Question number 21. The right answer is option C, to kill any bacteria present and prevent the contamination. Okay. Next question number 22. What is the use to maintain the 95% relative humidity inside the incubator? First option is the humidifier attached to the incubator. Second is a fan that circulates the humid air. Option C is the water placed at the bottom of the incubator. And option D is a separate CO2 cylinder. So the right option is option C, the water placed at the bottom of the incubator. Next move, question number 23. What happens if there are more petri dishes placed inside the incubator? So first option is more CO2 will be consumed. Second, the temperature will increase. Third, the water level for humidity will drop. And option D is the incubator will overheat. So which one is the right answer? So what is the right answer? The right answer is more CO2 will be consumed. Okay. So next move toward the question number 24. What should be done to prevent the contamination in the CO2 incubator? First is the regularly changes the CO2 cylinder. Second, routinely sterilize in the incubator at 90 degrees centigrade. Option C, open the incubator door frequently. And option D is the increase the relative humidity to 100%. So go through the question carefully and try to find out the right answer. So the right option is the routinely sterilize the incubator at 90 degrees centigrade. So next move to the question number 25. What is the maintaining a 37 degrees centigrade temperature important in the CO2 incubator? First, to keep the equipment functional. Second, it is the optimal temperature for human cells to grow. Third, to ensure the proper sterilization. And option D is to prevent the water from the evaporation. So try to find out the right answer. Why is maintaining a 37-degree temperature important in the CO2 incubator? So the right option is it is optimal temperature for human cell to grow. Okay. So why it is maintaining a 37-degree temperature important in the CO2 incubator? It is the optimal temperature for human cell to grow. Okay. So next move the question number 26. What is the significance of the CO2 a cylinder connected to the incubator? So first option, it provides the pressure for the incubator. Second, it's maintain the 5% CO2 concentration needed for cell growth. Option C is it prevent the overheating of the incubator, and option D is it ensure the incubator remain sterile. So go through the question carefully and try to find out the right answer. So the right option is option D. It maintains the 5% CO2 concentration needed for cell growth. Okay. So next move toward the question number 27. Which medium color indicates the cell are growing inside the petri dish? First option is the blue, second pink, third green, and fourth option is clear. So the right option is option B. It is pink. Okay. So in which direction can be micro manipulator system move? First option only in the X direction, second only in the Y direction, third in XYZ direction, and option D in a circular motion only. So the right option is option C in X, Y, and Z direction. Okay. So next move toward the question number 29. What is the typical resolution of the movement in a micro manipulator system as mentioned in the lecture? So first option is 1000 micron, second the 25,000 micron, and option C is 1 micron, and option D is 50 micron. So what is the typical resolution of movement in the micro manipulator system as mentioned in the lecture? Please try to find out the right answer. So the right option is option C, 1 micron. So next move toward the question number 30. What is the purpose of the indented in the micromanipulator system? So first option is to measure the temperature of the sample. Second, to apply the pressure or force on the sample. Third, to rotate the sample in different direction. And option D is to measure the electrical properties of the sample. So the right option is option B, to apply the pressure or force on the sample. Okay. So next move toward the question number 31. How does the control unit allow the users to manipulate the micromanipulator system? First option, by adjusting the temperature. Second, by controlling the XYZ movement through the knobs. And option C, by controlling the rotation of the sample. And option D is by monitoring the power supply. So which one is the right option? So the right option is option B, by controlling the X, Y, and Z movement through the knobs. Okay. So next move toward the question number 32. What is the role of the home button in the control unit of the micro manipulators? So first option, to return the manipulator to its default zero position. Second, to power off the equipment. Third, to measure the speed manipulation. And option D, to calibrate the system. So what is the right answer? So the right option is option A, to return the manipulator to its default zero position. Okay. So next move toward the question number 33. What What type of sample is typically tested using the micromanipulator and indenter setup described in the lecture? So first is the electrical circuit, second the biological tissue, third the metal plates, and fourth is the liquid samples. So what is the right answer? So the right option is the option B, the biological tissue. Okay. So next move toward the question number 34. Why are different modes available on the micromanipulator control unit? First, to change the resolution and speed of the X, Y, and Z movement. Second, to adjust the power supply to the unit. Third, to increase or decrease the temperature of the sample. And fourth, to calibrate the sensor attached to the system. So what is the right answer? So what is the right answer? Option A is the right answer, to change the resolution and speed of the X, Y, and Z movement. Okay. So next move toward the question number 35. Which company is mentioned in the lecture as the supplier of the micromanipulator system used in the lab? First, a MEMS technology. Second, the certain instrument. Third, the LabTech system. And option D is the microfab. So go through the question carefully and try to find out the right answer. So the right option is option B, the Saturn instruments. Okay. So next move toward the question number 36. So this is the last question. What is the functions of the work position button in the control unit? So first option, to save the power level of the system. Second, to save the return specific XYZ coordinates. Third, to monitor the temperature of the system. And fourth, to reset the manipulator system. So go through the question carefully and find out the right option. So the right option is option B, to save and return to the specific XYZ coordinates. Okay. So we are ending this session here. Next we will meet the 19th, I think 19th October, 7 to 9 p.m. Okay.